those. And if I fail to monitor, Simon will start waving his arms at me. I just want to start by saying a huge thanks to Simon, Vishnu, Hyung-wook, and Adam. Like, sink or swim, I am so grateful that you're doing this. In these crazy times, we have to be really creative about how to keep science moving forward. And so I really appreciate it. And I'm honored by the opportunity to come share some of this work with you all. This research was funded primarily by the National Institutes of Health. This project also received funding from the US Office of Naval Research. And I'm happy that we also have funding now to take this, these ideas in some related directions in collaboration with Simon and his group from the James S. McDonald Foundation. The experiments I'm going to tell you about were conducted by a very cool team of people, but the prime players here are Christopher Walheim, David Stwarczyk, and Michelle Eisenberg. I want to start by motivating this with an example. Suppose that you were to come to St. Louis and go to the ever excellent High Point Diner with your friend Leslie, and she were to order a big fat cheeseburger. You'd have now a set of features related to this activity bound in a spatiotemporal framework. It takes place at a particular time, particular place, with internal dynamics to it, like Leslie arrived before ordering a cheeseburger. And if you then returned to that location, oops, on a subsequent occasion with your friend Leslie, your brain might start automatically retrieving features of that activity and anticipating based on the dynamics of that retrieved event representation, leading to the activation of these cheeseburger features, which most of the time is great. It allows you to proactively respond in an adaptive fashion. But suppose she then orders a big old salad. That's going to lead to a prediction error. And so the key ideas I want to get out of this are that, in contrast to the way that we often study memory in the lab, in natural life, much of memory retrieval is an involuntary associative process that reflects queuing of previous event features based on the current context. And retrieval and encoding are not separate phases of an experience, but are constantly intertwined, and they interact to facilitate comprehension. And so the questions I want to take on here are, how is it that predictive comprehension processes trigger involuntary retrieval, and how do prediction errors trigger memory updating? The theoretical framework that Chris Walheim and I proposed to address this is represented here. It's called event memory retrieval and comparison theory, and it's represented by this diagram. And the idea is that at any given time, your perceptual systems present you with a bunch of features, and a lot of how we predict what's going to happen, or how our brains predict what's going to happen in the near future, is driven by those perceptual representations combined with our general knowledge. But some of it can be driven by associative retrieval of related previous experiences. So some of the features cue retrieval, and that can drive predictions about the near future. And the reason our brains do this is because most of the time it's great. Most of the time what happens is that things go the way they went last time, and you respond better because you retrieve that representation. And so you hang on to those representations and you use them again the next time. But sometimes things change in the world, and when that happens, you're going to experience a prediction error. And in the short term, that might be a bummer and lead to a disfluency in your behavior. In the longer term, it can actually promote error-driven updating. So there's a lot of interest currently in error-driven mechanisms of learning and memory updating. We're interested in these questions in the context of healthy aging and early Alzheimer's disease. And in that context, we've come to appreciate that aging could affect multiple stages along this complex pathway. So it could affect the integrity of your online event representations. I'm going to maybe focus a little bit on that one at the end. It could affect the computation of prediction errors. It could affect the use of prediction error signals as a trigger to memory updating, or it could affect the integrity of your long-term memory representations. So I want you to bear those possibilities in mind as we consider some data. Let me describe the paradigm that we've developed to try to answer some of these questions. In these experiments, people watch movies of a day in the life of an actor. The movie starts when she wakes up in the morning. It ends when she goes to bed at night. And each version of the movie can include 30 to 50 everyday activities. So this clip comes from about 2/3 of the way through the movie. She gets out of her car, approaches her house. Unlocks the door and goes inside. So after you watch this movie all the way through, we might tell you, now we'd like you to consider another movie in this person's life. And you're going to notice that on both of these days, you might see some of the same activities repeat. So about 2/3 of the way through, again, she gets out of her car and unlocks her door and goes into her house. Now, some of you right about now are noticing that that second time she unlocked the door differently than the first time. So whereas on the first day she unlocked the deadbolt, on the second day she unlocked the door handle. And notice that in this particular case, those two targets of the action are spatially dissociated from each other. That's going to come back up later. Here's a couple other examples. Right after she walks in, she takes out her phone and might lay it down on the keyboard of the piano or on the top of the piano. At another point in the movie, she comes from giving the dog a snack and then wipes up either with a paper towel or a towel that's draped over the faucet. So in one session, they watch a pair of these movies back to back and they're told to remember as much as possible. And then either a week or 3 days later, they come back into the lab and we give them a cued recall test. And I will just tell you, in case anybody's interested in doing these experiments, one of the really fun and frustrating things about constructing them was designing cued recall items that were open-ended but still focused people on the features that we were interested in. So we wound up coming up with cues like immediately after answering her phone, entering her home, on which part of the piano did the actor place her phone? And that's intended to focus people on, like, was it on the keyboard or the top of the piano, as opposed to telling us something else that might be interesting and true about her taking out her phone, but that wasn't relevant to our discrimination. Another item is, after placing her dog in the bedroom, what implement did the actor use to wipe down the kitchen counter? Potential answers could be paper towel or washcloth. So the design of this first experiment is going to have these two movies, day 1 and day 2. And in those two movies, you could have repeated items in which the same ending is presented, or changed items in which the ending is different on the second day, as in the example that I showed you. And then we also included control items that only appear on day 2. So for those of you who are aficionados of the verbal learning literature, this should look just like a proactive interference paradigm, because that's what it is. So you associate alarm with clock on day 1, and then either repeat it, or you have lock associated with handle on day 2, but something different on day 1, so you're going to get proactive interference, and then you have controls. Okay, and then a week goes by, and they get the cued recall test. This experiment included both young and older adults recruited from the St. Louis community. And let me walk you through a kind of complex set of behavioral results. So to start with, boy, I hope this is big enough on your screens. It's a little small on mine. But to start with, I'll read these just in case the labels are too small. You've got your control items here, and compared to those, the repeated items, surprise, surprise, are remembered better. But what about the changed items? So these are the items that would produce interference in a proactive interference paradigm. And what we find is they do not, in fact, produce proactive interference. We see instead proactive facilitation. And that proactive facilitation is conditioned strongly on whether they can correctly answer for us after answering the cued recall question. Can just tell us whether it changed or not. So if they recollect that it changed, they're quite likely to be able to tell us what those change features were. Whereas if they falsely recollect it as being a repeated item, they're quite likely to misremember the features. And in fact, what they do in those cases is intrude features from day one. So these are intrusion rates for the same items. Now, these data all come from younger adults. If we add in the older adults, what you see is that overall, the older adults remember a little bit less, and they show less of this proactive facilitation. It turns out that that is accounted for by the rates at which they can correctly recollect the fact that it changed. So it may be a little small on your screen, but the size of these balls is telling you the proportions, and you can see that the majority of the younger adults are able to recollect that the changed items were changed, whereas only the minority of the older adults recollect that. When they do, they're seeing a similar benefit from being able to do so. So encoding the fact that an item has changed is associated with remembering which feature has changed, and this leads to proactive facilitation. Older adults show less proactive facilitation and less remembering of change. For the next study I want to describe, we wanted to zoom in on the prediction part. And we wanted a non-invasive measure of prediction so as to not mess up the ongoing encoding and comprehension processes, and most importantly, the spontaneous retrieval and prediction processes. So what we did is we took advantage of irregularity in human and other animals' eye gaze behavior, which is that whether we're acting in a situation or just passively observing it, we tend to look ahead at the objects that the actor is going to interact with. So if I'm about to reach for a coffee cup, or if I'm watching somebody reach for a coffee cup, my eyes are going to land on that coffee cup ahead of the hand. And so that's an online indicator of prediction that we can use to assay what people's brains are predicting about what's going to happen next. We can only use items where the locations of the action targets are spatially dissociated here, so we got a few fewer items. So we cut the control objects in this experiment. The rest is about the same. This is all younger adults also. So here's just an example highlighting those different spatial locations. So we can draw these interest areas and measure prediction as the proportion of time that people spend looking in the interest areas. And each of the activities differ in length, so we're going to warp time into two sets of 10 equal time bins. So we've got 10 bins from when each activity starts up to the point where these two versions diverge from each other. Then we got another 10 bins that go from that point to when the hand touches the object. So what we're seeing here is on day one, the proportion of looks to the target. And so here's our two phases of the activity, up to the divergence point and then divergence point to the contact. And you can see that well before the contact point, people are starting to look at the target object. And it's about the same for the repeated and changed items, which is good because on day one, they're the same. It's only on day two that we're going to see differences between repeated and changed. For the repeated items, what we see is that they start looking at that target object way early. For the changed items, we don't see them looking early, but they do catch up and still look predictively. What are they doing in that early phase? Well, if we look at the looks, so if this is or actually this is where she reached on day two, what they're doing is they're looking with some frequency at the thing that she had reached on day one. So this is exactly the hypothesized memory-based prediction error. So if they're retrieving a representation of this and it's guiding their looking, this is the error that we're going to make. And this is exactly the error that we hypothesize would be causally related to memory updating. And so to get a little bit stronger test of this, I want to be frank that this has to be a correlational design, because we can't push people into the make the prediction error or don't make the prediction error condition using this design. But to get a little bit stronger test, we backsorted their memory, and in particular their ability to recollect that the item had changed. We backsorted the looking data based on whether they could correctly recollect that it changed. So that's what I'm showing you here. And what you see is that on those trials that they later answered correctly when making the memory judgment, they were more likely to have made this error during encoding of day two. So this prediction error during day two is associated with superior memory a week later. So in this experiment and one other, we found that memory for similar recent event produced errors in predictive looking when things changed. And these errors were associated with better encoding of the changed ending. In other words, prediction errors were associated with memory updating. Last experiment I want to tell you is a pattern-based Fmri experiment that was designed to zoom in on the reinstantiation of those episodic representations during day 2 viewing. So to do this, we did fold a little bit on the naturalism and added some pauses into the movies. But the design is basically the same. So again, we don't have control items to maximize power, but they're going to watch a day 1 movie and a day 2 movie with repeated and changed items. They're going to take a memory test later. This did have younger and older adults. But what we do here to be able to measure pattern reinstatement with a hope of having enough signal is after each of the critical activities during the day 1 encoding, we pause the movie just so that they know what we talk about. When on day 2, we pause the movie at the divergence point and ask them to think about what was going to happen next based on their memory. So they need this pause to know what they're supposed to be thinking about here. Then we show them either the changed or repeated ending. And then we ask them, were you able to successfully retrieve? That's a question about this phase. And then we ask them, did it change or not? Which is a question about what happened here. First, I'll show you the memory data showing that we replicate everything I showed you in the first experiment. So changed items are a little bit worse than repeated items, and it totally depends on whether they're able to recollect the fact that it changed. Next thing I just want to mention briefly is the mass univariate signal that you observe when people hit this point in the paradigm. So, oops, sorry. So here, when you see that post-divergence start, and it's not going the way it did last time, there's a big uptick in the mass univariate area throughout a lot of the brain, including the hippocampus and surrounding structures in the medial temporal lobes. But what we're really interested in is what's going on here, and we're interested in the patterns that are being reinstated there. So to measure that, what we did is took a large number of regions of interest from a parcellation by Schaefer and colleagues, and for each of those little parcels, we extracted the pattern over space, averaged over time during that 12 seconds. We averaged the 12 seconds when they were encoding the ending of day 1, and then the 12 seconds when they were anticipating the ending during day 2 viewing. We did that for all 45 activities. And then we can compute just the correlation between, say, encoding day 1 and anticipating the ending on day 2 for the same activity, or matching like the first activity with the 45th activity, right? So what's the correlation between when you were encoding the first activity, but then you're retrieving the 45th? And if they're retrieving activity-specific information, instantiating activity-specific patterns, then you ought to see stronger pattern similarity on the diagonal than off the diagonal. So our score for how strongly people are instantiating these patterns is just going to be the diagonal minus the off-diagonal elements. Okay, so this is just illustrating all these regions in the Schaefer atlas and this medial temporal lobe region of interest. And across these large number of regions, most of them, if you just ask, is there significant pattern reinstatement? It's amazing to me, actually, how much of the brain shows significant event-specific pattern reinstatement. It seems to be a ubiquitous signal in lots and lots of brain areas, including the areas that we were interested in this network that's referred to sometimes as the posterior medial memory system by Trent Rangnoff and colleagues, or by the contextual association network by Adrian Gilmore and colleagues, and also in the hippocampus. And this is just showing quantitating, averaging over those 2 large swaths. For the posterior medial cortex, both younger and older adults show significant reinstatement. For MTL, it's significant in younger adults only. Now, how does this degree of pattern reinstatement relate to behavior? The analysis I'm going to show you next is a between subject analysis where we're going to look at each individual's overall reinstatement score compared to 3 behavioral measures. So we've got the posterior medial cortex on the top, medial temporal lobe on the bottom. On the x-axis, we've got our measure of neural reinstatement, and on the y-axis, we've got the degree to which they reported that they were able to reinstate when we asked them that question during day 2 viewing, then their objective day 2 recall performance, and their objective change recollection performance. Let's start with the younger adults. For the younger adults, you see these robust relationships such that people who show more neural pattern reinstatement have more subjective reinstatement. That is, they're showing metacognitive calibration. When the people who thought that they were retrieving more, their brains are showing more reinstatement. This is true in both the PMC and the MTL. They're showing stronger day 2 recall in both regions, and they're showing stronger performance on the judgments about whether things changed. The older adults are really interesting. On the objective measures, they're worse overall, and they're not showing a very strong relationship for either of them. And for the subjective measure, they're showing this dramatic overconfidence, where the older adults are almost on the ceiling, telling us, yeah, sure, I retrieved successfully, even when their brains don't show evidence for that, and their behavior doesn't show a lot of memory sensitivity. So there's a metacognitive gap there. So pattern reinstatement in the PMC and MTL was associated with self-reported reinstatement. Pattern reinstatement in the PMC and MTL during event changes was associated with detecting those changes, and pattern reinstatement was associated with better recall. This is all in younger adults. Compared to the younger adults, the older adults showed less neural pattern reinstatement and showed weaker associations between reinstatement, subjective experience, and memory. So one way of thinking about this in terms of this EMRC model, this is going to come back to this question about where the deficits might lie, is that maybe there's a reduced ability to form that online representation based on memory, which is going to mess up your ability to do event-based prediction, which in turn is going to reduce the prediction error signals, which is going to, in turn, could mess up your fail to trigger memory updating. Now, that's just one possibility. I want to be clear that I don't think that these data rule out some of the other possibilities that I laid out, but it's an intriguing one that I think is worth following up. So zooming out, during comprehension of a given activity, memory for an associated previous activity can drive predictions about how things are going to go. And most of the time, this is adaptive. I just want to highlight the reason that our brains are built this way is because it's good to have representations of related previous events and use them to drive your predictions. But when things change, this can be a short-term hiccup. This can lead to a prediction error, which will lead to disfluent performance. But in the long term, it can lead to event model updating, integration of the prediction error and the prediction and the error into that memory of day 2, leading to what Hintzman called a recursive representation, which can drive improved learning and memory. So I'll stop there, leaving a little time for discussion. I just want to, again, thank the larger group from the lab that was involved in carrying out these studies and helping us understand what they might mean. And thanks for your time.
Awesome. Thanks for that, Jeff. Lots of claps. So are there any questions?
I've got a question if no one else has one.
Yeah, go ahead.
Just thinking about the aging data, I know you don't really want to commit to a particular theory of it, but I was just curious. One of the things I found really intriguing, Michael Ramskar has a model of aging that assumes that there's no aging deficit anywhere.
Yeah,
it's just that older adults have more memories and then they just have more interference. I mean, it strikes me that you could kind of similarly have an impairment in prediction error for that reason, which would kind of cycle through the model and make some of the same kinds of predictions. But I don't have as strong of intuitions here. So I was just curious if that might be another viable explanation of the data or not. And yeah.
Yeah, that's an intriguing one. I mean, you'd have to model it. I think how it would turn out would totally depend on the assumptions that you built in about the similarity, structure, and strength of context, temporal context in biasing retrieval, right? So, you know, if you've got like these 2 trips to the High Point Diner, and those are much more similar to each other than all those other memories in your data bank, and also, or if temporal context drifts at a reasonable rate and has a big effect on guiding retrieval, then that number of memories story probably won't do it, because it probably can't overcome those things. But if you think that there might be other trips to the High Point floating around that, or you know, related restaurants that really could get in there, then you've got a shot. Yeah, that's
Right. Actually, yeah. So maybe I just thought of another maybe follow-up question, which is one of the ways that Michael Ramskar, I think, distinguished his predictions is he found with really novel word pairs, really distinctive word pairs, you find almost no aging deficit because they don't really have any interference at that point.
Mm-hmm.
You can get older adults to look like younger adults. Have you found any kind of in your work with like really unique events? Do you find less of an aging deficit or anything like that?
We don't have any data that speak to that. And I don't know of anybody who's tried to pursue like extreme distinctiveness in naturalistic stimuli. I don't know, somebody else on this call might, but I don't know of any data like that.
Got it. Thank you.
Yeah, thanks, Adam. Those are great.
So are there any other questions?
Can I ask a question, Simon?
Go for it.
Wonderful talk, Jeff. So I was wondering, with the Fmri study, is there a way to establish that the older adults have sort of a similar representational fidelity of the event structure, and that it's just sort of a deficit in reinstatement? So sort of the
in fact, no, great. Yeah. Sorry, I didn't mean to cut you off, but that's such a great question.
But you get what I mean, yeah.
Yeah, and I think I just want to be clear that like the thing that got us started on this was Thad Pollock's work on de-differentiation and reduced representational fidelity. So whatever the pathway, as long as that thing that you're reinstating is fuzzy or sloppy, that's going to kill you. That's the basic idea. Yeah.
Okay, thank you.
Okay, cool. So we might switch now. So thanks again, Jeff, for a great entry to the conference.